Professor Utpal Tatu. Professor Utpal Tatu is a professor of biochemistry in Indian Institute of Science and dear colleague. And Utpal is going to speak on the proteogenomics of SARS-CoV-2. Utpal, can you kindly turn on your video? And I would kindly request to restrict the talk to 25 minutes so that we can have 5 minutes for the questions.
Okay. Thank you, Professor Dipshika, for a kind introduction. I'd like to thank the organizers for giving me a chance to present at this conference on COVID-19. Are you able to see me?
Sir? Yes,
are you able to hear me and see?
Yeah,
okay.
Yeah, we are able to hear you and see you.
So if it's okay, I'll turn off the video. I think it's just
sure.
Because I've shared my slides. I hope you can. My slides are.
Yeah, it's visible. It's full screen. Yeah.
Okay, thank you. Thank you. So my presentation is going to be about proteogenomics of SARS-CoV-2 using NGS and high resolution mass spectrometry. And I'm at the Department of Biochemistry at Indian Institute of Science. For the interest of time, just a minute here, I'm going to actually give my acknowledgements first in case I run out of time towards the end. Just want to acknowledge all the students who rose up to the occasion and participated in this study, which we have done in the institute. Graduate students who were engaged in other research, but they dedicated some time, as well as project assistants, postdocs who sort of rose up to the occasion. Sheetal, Satish, Vinitra, Advait, Sujit, and Aishwarya Bhatt, who provided the bioinformatics support. Clinical collaborators all over Bangalore. We had a lot of support from clinical early on, of course, also from NIMHANS, Dr. Ravi, but also from Dr. Manjunath, Dr. Vishal Rao, and other clinics in Bangalore who supported us. I really want to thank the Institute Virus Research Committee for evaluating our proposal rather swiftly and advising us on what's the best way to move forward. And I also want to thank Institute Human Ethics Committee for putting together a very efficient mechanism to evaluate proposals to allow work being done in the campus at a time when nothing was very clear. That's what's right and what's wrong. So I appreciate their effort and initiative and being swift without having to stall anybody's work. I'm also thankful to ICMR Consortium to study host responses to allow me to communicate with them and work with them. I'm starting my presentation with this sort of a historical slide, which is where I come from. My postdoc at Yale several years back, it's an old story now, 24 years, was predominantly on influenza virus. And I was looking at influenza virus protein maturation and trafficking in the host cell, endoplasmic reticulum and Golgi. So that's really where my expertise was during 7 years of postdoc. But when I came back to India, I decided to switch and start studying infections that were prevalent around me at that time, malaria and other protozoan infections. But now I think COVID-19 presented an opportunity for me to that maybe I can use my expertise in a way that may helpful. So my long-term goal really is to connect my basic expertise in studying cell biology of virus protein maturation. And I'm showing you this here, I think some of the events that have captured my postdoctoral work, which are now textbook knowledge. This is from Lodish edited molecular cell biology book, wherein my work showed how different co- and post-translational modifications of viral glycoproteins help in its folding and assembly in the endoplasmic reticulum, and how that's linked to trafficking and then virus assembly and egress out of the host cell. I'm hoping to apply similar principles in the years to come on COVID-19, because I think this is going to be very interesting. So the long-term goals of my COVID-19 interests are really to understand viral glycoprotein assembly in the ER, and thereby hopefully cell biological level, we may think of novel intervention strategies. And then the low-hanging fruit really was to begin to look at proteogenomic analysis of COVID-19 based on the expertise that I have in the lab on mass spectrometry and generally on the big data science aspects of biology. I'd like to comment here that in the current scenario, my first two goals sound very irrelevant because we are in a fighting mode. It's a pandemic time, and we are far from in a safe area. But I do believe that nobody has had enough time to really dive deep into the biology of the virus. Professor Soumitra Das, of course, is one of the experts in virology in the country and at international level. He probably understands this and he's doing a wonderful job there. I saw his wonderful talk. But I do think that in the end, the long-term solutions will lie in diving deep into the biology of this virus, which includes some of the cell biology angle, which I hope I'll have time to explore. I'm going to skip this first slide because we've seen this several times today, basic structure and of COVID-19. But I just want to emphasize here that it's one of the biggest genomes for positive strand RNA viruses compared to other viruses, and as a result, a lot of things that are happening in the genome, a lot of proteins being coded, and I think we are just beginning to understand what's going on with the molecular jugglery at the genome and proteome level. I would say at proteome level, we understand very little, and which is the direction in which we want to head. So I'll skip over this slide very quickly to come and say that while the genomic organization has told us all the genes that the virus is capable of coding for, and there's a highly under discussion, and again, a target for diagnosis as well as some of the intervention strategies that people have come up with. But we know very little about these proteins really at the protein level. So of course, all these genes which are listed here as protein and nucleocapsid, membrane, envelope, probably the audience knows more than me because we hear about them not only in scientific publications, but also in newspapers and WhatsApp groups. And then of course, the wonderful bioarchives, which makes everything accessible so rapidly. We know the first 4 or 5 genes that I listed quite well. And a lot of work has happened on these, even in this the mutant form of the virus, which can be expressed in the cell culture cell lines. And people have begun to look at some of these proteins and particularly spike glycoprotein. We know a little bit less about the non-structural proteins of the virus in terms of what they do and how they do. But the third category, the accessory proteins, we know least about. And these are the open reading frames. And I just want to point out that some of these open reading frames are really enigmatic because we only know them as genes and there is really no information at the protein level. And this is where my excitement is, that can we actually try and dig into the proteins which are expressed by the COVID-19 in the host when it carries out infection. Again, I think we have some predictions. We have structural predictions of many of these viral proteins. We, based on wonderful bioinformatics that has gone on, we understand what fold they may take, what function they may carry out. But I think we are far from anywhere close to understanding the reality. I'll just give you an example of ORF10, which is again enigmatic protein. To my knowledge, this particular protein very nicely coded in the genome of the COVID-19, but very few people have actually seen the protein being expressed in the host. I don't know of many reports which talk about it, and I will not rule out the possibility, but there may be something that I missed. But similarly, ORF10 is not the only example, but there are other genes whose protein products remain elusive, and as a result, we don't precisely know their function. I mean, I'll emphasize about ORF10 again because it shows homologies to some protein in the database, but we really don't know what it does because it's I mean, I won't belabor this point more than this, but clearly. Hello?
Utpal, yeah, we can't hear you.
Yeah, I think suddenly the Wi-Fi gave way, but I'll switch back. Somehow there was a. Sure. I'm in the department only, but now sure. I think it's back.
Yeah, it's back. It's back.
I apologize for that. So I think the message that I'm emphasizing here is that in the times to come, more and more efforts will have to be put on examining proteins, because there are some unique proteins, not necessarily only SARS virus, but also for SARS-CoV-19, which may actually have secrets of its unique biology and the unique challenges that this virus has thrown at us. So really, I am a big ambassador of the clinical proteome. In the past, I have spent a lot of time examining the clinical proteomes of other infectious agents, including malaria trypanosoma and many other protozoan infections. But I think in the times of COVID, again, this is going to be very important. Now, some quick points about why clinical proteomics. Because I think in some ways, even at a very, very low hanging fruit level, in terms of diagnostics, examining a protein is going to be of tremendous advantage because it provides information about proliferation. You know, in the world which we live in, where there is this question about false positives, false negatives, I think protein level diagnosis is going to be really nice. People are doing antigen tests, but I think there is tremendous scope to improve this. So I think moment you see a protein coded by the virus, at least you are sure that the virus is replicating and producing the gene products, and it's an active infection. Protein copy numbers being higher, multiple copies, multiple proteins, better sensitivity. So there is a scope to develop better methods of protein-based diagnostic tools for COVID, and I think we haven't explored enough at this point, apart from the antigen-based test, which is available and which is highly questionable, unreliable. Functional interpretations may become possible. Of course, when you see a protein, you see its post-translational modifications, you begin to understand the biology much better. And then finally, most importantly, I think when we do proteomics, we will see host responses. And that's going to be fantastic. The different categories of virus-infected patients being, the most sort of asymptomatics to moderate to severes and the most serious infections. How do the host responses? I think Professor Soumitra nicely pointed out that this is where the holy grail is. We really want to understand the differences. And then at genomic level, a lot is being done, of course, but I think this will eventually we will need to understand what are the responses triggered by the host. So with this broad perspective, we embarked on examining proteogenomics, of course, at a time when we were just beginning. And we are really at a very early stage. So none of my work is published. It's under publication, under submission, under review. So I hope in the times to come, it will come out in the public domain. But we decided that with whatever access that to material that we had, and which was very little, as all of you agree, we decided that let's use the tools that we have in the lab. And that meant that we did the throat swab analysis that's being carried out for testing. We use the same samples for, on the one hand, like all the other wonderful studies that were described today, to examine genome sequences. And on the other hand, we also dabbled into, can we now begin to enumerate proteins produced by the virus? I'm going to cut the first part of my talk short because the champions have talked about it, and I'm really a newcomer in this field. So genomes, I'm just putting samples of genomes that we sequenced to show you important highlights. And this corroborates with what most of the findings that actually have been discussed this morning today. I think that there are various single nucleotide polymorphisms, mutations, the clades, and all of that is very nicely agreeing with all the data that's being shown at a much, much bigger level. But some of the samples had very interesting surprises for us. And particularly on the top panel of this slide, I'm showing you the common mutations that were embedded in the genomes some of the genomes that we sequenced. And bottom line, what amino acid changes, if at all, they converted it. I'm not going to belabor this point too much because much nicer and elaborate and exhaustive studies have been presented. But the summary of this work, and I kind of, while listening to the studies done by others, I kind of filtered out and only highlighting points that I think are unique to what some of the samples that we have studied. The point was that, like everybody else, we found the G clade, we found the GH and GR clade, but there were some interesting surprises that we found certain clades which did not belong. And these were others where certain mutations which are not yet fixed in a particular clade, we were able to find. I'll be happy to share with you with anybody interested, but then we all talk about the G614G mutation, and that's something that did not escape us. It was obvious that we also had it. And more interestingly, as I said, some of the mutations, predominantly most of the sequences that we looked at belonged either to GR or to GH clades. As I said, this part of the work, I'm going to move fast because I don't have anything really significantly different from the wonderful studies that were presented earlier today, except that I would like to emphasize here that the sample genome sequences that we looked at, actually, two of them were very close. In fact, very, very close to the Bangladesh isolates. So we are having some sort of a crosstalk with that part of the world here, and in turn they were close to the European isolate, which again agrees with what others have shown. So clearly, I think some new data was coming out, and I do believe that every sample, every genome that is being sequenced will provide valuable information, even though large number of genomes have been sequenced. One of the things that came out of our little study, a tiny drop in the ocean that we could contribute, was that the mutation rates are really going out fast. This is from previous published studies showing that 7 to 8 mutations are on the average, and these are typically present. I think India is infamously the house of highly mutating virus, and as a result, I think we found, in fact, that our study also corroborated that the sequences that we found had more number of mutations than even the 7 and 8, which have been the maxima as published in other studies, because some of the samples that we looked at actually had many more mutations, altogether 10 to 11 mutations, deviating from the standard clades that we have studied, and we are now scratching our head to understand why so and what is the underlying mechanism of this differences. But at this point, I want to move away and jump into the proteomics, because once again, I don't want to raise your expectations because these are early studies, and we've just begun to look at some data. And I must confess that there's already some data in the literature, but compared to genomics, there is relatively less. And I think there are some challenges unique to examining proteomes of the virus from the patient. And I'm going to share that with you because I think many of you, my colleagues, are much better placed than I am, because there is access to much bigger facilities, wonderful machines, and I think this is going to be very useful, and I'm very, very eager to collaborate with people who have much bigger machines and better technologies that is accessible to them. So we again relied on the nasopharyngeal swab, inactivated them in molecular transport medium, TCA precipitated, resuspended in SPS, and just to cut the long story short, we are definitely experts in protein preps. That's what proteomics is all about. And we sort of played around with various protocols and came up with something that gave us some hint of the proteome. I must emphasize that it wasn't easy to examine viral proteins in the midst of highly abundant host proteins in the throat. Imagine taking a throat swab, epithelial cells from the throat, and then looking for viral proteins. They are definitely going to be in femtomoles or picomole amount, of course, depending on the virus load. But in our experience, even with the lowest CT value samples that we got, it wasn't really easy to, the abundance of the viral proteins was always on the wrong side of the abundance in the depth of the proteome was always in favor of the epithelial proteome, the host epithelial proteome. The point that I want to emphasize here is that for us, both were interesting. On the one hand, detecting needle in the haystack, looking at a needle in a haystack, a viral proteome. But on the other hand, the host proteome did carry important significance in terms of examining, is there a unique component of the host proteome away from the uninfected subjects, individuals who are RT-PCR negative, compared to them, were there unique proteins in RT-PCR positive samples? Again, this data is under submission, and I hope to hear some good news in the days to come. But just to summarize, these are the sort of heat map of which are the proteins that we rapidly found, which we easily found. And of course, this is not a representation in the sense that this is by no means sort of a, I'm sure somebody else will do better than us in terms of, but these are really the first studies, the first proteomic data that came out of our instrument. And we found some very interesting proteins coming out here. In addition to the nucleocapsids and RDRPs and NSPs, we also found enzymes and which are actually now beginning to tell us something new. In fact, the E's and N's and structural glycoprotein. Again, I think. I apologize for this. I think it's a Wi-Fi issue, but I think luckily it is short enough, so I'm able to switch back. Thank you for your patience. The point that I want to emphasize here is that there were, there's something new to think about beyond just E gene and N gene and, the RDRP. There are many other gene products that we are beginning to see. and very abundantly expressed peptides we were able to see about 8 to 10 peptides oftentimes, but sometimes as low as 2 peptides, but very high confidence peptides. I will not go deep into the data analysis at this point. I want to give you a gist of it, but to say that there is fascinating potential possibility of examining the COVID proteome. These proteins belong to various intracellular pathways. I want to highlight here that the infected host cell also had some interesting story to tell. In addition to looking at viral proteins, we found large number of proteins from the host origin. In fact, difficult to hide them, and in fact, difficult to deal with them because they were suppressing major peaks from the viral proteome. But we found that RT-PCR negative and RT-PCR positive, when we compared them, we had, of course, hundreds and thousands of proteins, but there were many proteins that were unique only to RT-PCR positive patients. So subjects who had tested RT-PCR positive for COVID-19 had protein expression profiles that were unique, that were absent in those which were RT-PCR negative. And that, I think, was significant, and that we began to look at what pathways they belong to. And now some functional data is emerging out of it. Again, I will not belabor this point because right now I want to emphasize more on the big picture. The data is under submission, and I hope to hear from them. But this is really the sort of summary of the kind of data that's coming out based on the proteomic signatures that we found, in addition to the viral proteins of different gene products. Shown here in red, we found gene IDs shown all over the host cell are the ones belonging to the host proteins, and these are in fact the ones which we have highlighted are unique to the infected cell. So I mean, all of these proteins that are shown in this figure are actually at this point are unique to the host cell. Now I want to put a disclaimer here that I'm not ruling out the possibility, because I think in the end the big data science depends on the sample size. As we examine more and more samples, we may have to filter out some of these proteins, because as our capacity to go deeper into the proteome, we may find some of these proteins also being expressed. And the other disclaimer I would like to make is that this is, of course, a non-quantitative data that I'm presenting to you. We have done quantitation only through spectral count analysis, but not through the ICATS and ITRAC analysis. The data is, I'm not sharing at this point, but we have done some of this analysis and some quantitative picture is now emerging. But these proteins were absent or present. So that's the difference that we are looking at currently with the limited data that we have based on the proteome of the COVID-19 virus in the host cell. I want to leave a few minutes for discussion. So I'm going to wrap at this point and thank the organizers again for your kind involving me in this conference, and happy to answer any questions that there.
Thank you, Professor Utpal Tatu, for the wonderful talk and giving us an insight about the proteomics with the SARS-CoV-2. Now we have very good time for a couple of questions. The first question I would like to ask is from Rhythm. He wanted to ask whether it's a wonderful work out of curiosity. You want to ask that which regions you got were highly mutagenic for SARS-CoV-2.
So where did we find maximum mutations? Is that what the question?
Where do you find maximum mutation? Yeah.
So again, it was not very different from what others have shown, but yes, we did find in our data, N gene was one of the sort of foremost genes where we found larger number of mutations compared to others. Of course, S glycoprotein, as Shaumitro highlighted already, was again a place where there were quite a few mutations. Altogether, 11 mutations were commonly found, and in N alone we found 6 mutations. If I'm answering the question correctly, but correct me if I'm sort of going tangentially.
Thanks, Utpal. The second question is that are these SARS-CoV-2 proteins, they're secreted or shed from the dead cells? By Shashank.
Fantastic question. That's the functional data. I think that we are beginning to ask that again. So I didn't go into the details. So we were actually able to differentiate between intracellular and extracellular proteins. Now, extracellular would include even from dead host cells, the shed proteins, and the actually bona fide secretory proteins. So we do have demarcation between the two based on the sample preps that we did, based on whether we took the pellet or the supernatant. We have all of that data. I have not shown that data in terms of shown the separate. So currently the proteome that I showed included both intracellular as well as either through host cell lysis or through secretion. But yes, we do have separate data now. Very good. That's really the line along which we would like to think to begin to make sense of some other at functional level.
Professor Kumar Somsundaram asked a question regarding the correlation between the CRP in the serum. Is it correlated to the severity of the COVID-19 disease? And did you also find the same?
Professor Dipshika, what is CRP, right? You are talking about CRP.
Yeah, C-reactive protein. Yes, yes.
Right. So no, we didn't have. So far, as I said, we didn't have any quantitative data comparison, especially for CRP, because we would have to compare CRP levels between normal subjects versus COVID-19 positive, which we haven't done. For me to comment about this acute phase protein, of course, is very, very relevant, but I don't have conclusive data to comment on that. I want to make one more comment, if I may.
Sure.
Very
sure.
quick.
Please. Yeah. Yeah.
If there is no further question, I think what we are doing currently really is to now set up a sort of a quantitative MRM based assay, which is now almost ready in the lab, which is really going to be field applicable MS based diagnostic. That's going to be, I think, very useful, because there you don't need this big instruments of the kind that we use for global proteomics in this case, but MRM based diagnosis is where we think that the future of, we can conjecture miniaturized mass specs at the hospital, at the airports, where quickly, taking a matter of few minutes, you could do a sample for quick analysis. And that's really where the future of mass spectrometry based diagnosis is going to be.
Sure.
Thank you for giving me that.
Yeah. Thank you very much.